We are going to go ahead and get started with session 2 of the meeting, which is the lightning talk session. My name is Camille Pottinger, for everyone who has not met me yet. I'm with the Epidemiology and Genomics Research Program here at NCI, and I will be moderating the session along with Heather Eliassen, an associate professor from the Harvard School of Public Health and a steering committee member. We will be hearing from 5 speakers who will be giving 5-minute presentations on either a new idea for a consortium project or share results from an existing project. Please feel free to leave your questions in the chat, and we will have a discussion at the very end of all 5 presentations. I would now like to introduce Andriana Halavati from the Vanderbilt University Medical Center, who will present on a newly approved consortium project, the Appendiceal Cancer Consortium.
Well, thank you, Camille, for that lovely introduction. It's a pleasure to have the opportunity to present today on, as you mentioned, a project that was just recently approved and is getting off the ground, entitled APEC, or the Appendiceal Cancer Consortium. If you can please go to the next slide. Thank you. So one of the striking observations that was recently published earlier this year is that the incidence of appendiceal cancers have increased approximately 232% over the last 2 decades, with rates rising from older to younger generations, as you can see in the figure depicted here. However, one of the striking things to note is that over this time period, rates of appendectomies, which usually leads to the incidental diagnosis of appendiceal tumors, have remained stable over this time period, really leading to questions as to what causes or underlying this rising disease burden of appendiceal cancer, including among young patients in particular. However, as a rare digestive system tumor with an incidence rate of about 0.12 per 1 million person years, it's challenging to conduct studies using an individual cohort approach. And as such, we thought the NCI Cohort Consortium was uniquely suited to be able to overcome this limitation and make advances in appendiceal cancer, which to date have remained very limited. If you can please go to the next slide. And so that leads to the overarching objective of APEC, which is to understand risk factors, etiologies, and prognostic factors of appendix cancer, and to utilize this knowledge to reverse the increasing disease burden, as well as to inform clinical, molecular, and population level features that contribute to appendiceal cancer disparities, which my group has published on and continues to focus on. And so to do this, given the rarity of this malignancy, we proposed to leverage a nested case control study design and began reaching out to cohorts this summer after our project was approved by the steering committee. Currently, we have about 25 international cohorts within the NCI Cohort Consortium that have agreed to participate, which amasses to over 1,200 cases of appendiceal cancer presently identified and counting, as some cohorts are still querying their numbers. The intent is to match appendiceal cancer cases to non-cancer controls within cohorts at a 10-to-1 frequency. And also, given the emphasis recently on the distinct features of appendix cancer versus colon cancer, to also match 2-to-1 to right-sided, left-sided colon and rectal tumors within cohorts to be able to make additional comparisons in distinct disease features between appendiceal and colorectal tumors. If you can please go to the next slide. So I've listed here some of the participating NCI cohorts that have confirmed approval, but one of the opportunities at this platform is to really kind of reach out once again, in addition to individually following up with cohorts, that any cohorts with appendiceal cancer cases we are interested in still including in APEC as we begin to take the next steps For the consortium, individual cohort proposals and data use agreements have already begun to be drafted to move this project forward, and an R1 grant application is planned to support APEC infrastructure and to address some of these initial research questions for spring of 2021. And so I've included my email here at the bottom of the screen. If any cohorts are still interested in participating, to please reach out to me, and I will follow up with ones that I've previously spoken with or reached out to as well, as we're thrilled to have this really unique opportunity to make significant advances in our understanding of a rare malignancy via this cohort consortium approach. And thank you again for the opportunity to present.
Thank you, Adriana. I would now like to introduce James Lacey and Emma Spielvogel from the City of Hope, who will present on how to add free and secure interactive data visualizations to your cohort or project's website.
Thank you, Camille, and hello, everyone. It's good to join you this morning. Could I have the first click, please? Excellent. We've heard data visualization and these sort of key questions that come up in every cohort be raised already a couple times this morning. Every project starts with a couple of key questions. You know, does a cohort have specific data on this factor? How many of X outcomes or X biospecimens does a cohort have? We found that data visualization is a great way to answer these questions. As a lot of us have seen, data visualization is a priority for the NIH overall data science plan. And we've found that it can be implemented in cohorts and projects using a number of free and user-friendly tools. Could I have the next click, please? Within the CTS, we started using visualization for some internal analyses first, and recently we expanded that to include some interactive visualizations on our website. We want to share them with you today because we think some of these templates could be reused by your cohorts, your project, and in particular, what's nice about them, they're free, and because they present summary data rather than individual data, it sort of tackles and avoids that data security question. One last click, please. Before I turn it over to Emma, who's going to show you some of the details, I'd like you to have in mind, you know, as you see these visualizations and what you've done, try to think of similar data in your own project or in your own cohort and think, you know, for that particular project, could some of the things that we've done, you know, be reused in your particular project? So Emma, I'll turn it over to you for the demonstrations.
Thanks, Jim. If we could go to the next slide, please. So first, I'll be walking you through two of our interactive visualizations from the CTS data page on our website. So CTS participants have completed 6 questionnaires since 1995, and researchers often come to us wanting to know what topics were asked about on those questionnaires. If we could go next to start the video. So on this visualization, the bubbles represent the different topics on those questionnaires, and the size of those bubbles represents the number of questions that were asked on each topic. And when you hover, you're seeing a little bit more detail. The color corresponds to the questionnaire. So if a researcher wanted to know what did you ask about on the first questionnaire, they would click Q1 like in this demo. And now the view is filtered to only show what was asked about on Q1. So you see a lot of female health history, diet, pregnancy, etc. Then we can reset the view. And let's say instead a researcher prefers to view this by topic. Here you're seeing all of the topics. They can scroll through and pick their topic of interest. Here it's going to be diet. And you can see all the questionnaires that asked about diet and how many questions. So we see a lot on Q1 and Q4, and then a few on 2 other questionnaires. And they can always reset this view and search for something else. If we can go next, please. We also have a visualization for our biospecimen collection. Next, again. So in the past, we had struggled to display complex information about our biospecimen collections like this in a way that was useful for researchers. And this visualization has helped us to do that. So here you're seeing our different biospecimen projects over time. And color corresponds to the different sample types. So red is blood, this teal color is saliva, and so on. And in addition, hovering over the chart will give more details. So things like the collection dates you're seeing there. And down below, you can see the specific counts of participants who gave each type of specimen for each collection project. Both of these visualizations are a really big time saver for our internal team, as researchers can answer a lot of their initial questions themselves without requiring us to go back and forth via email to answer these questions. Next slide, please. This last visualization is on publications using CTS data. And next again to start it. Here you're seeing the number of CTS populations by topic on the left and then by time on the right. For example, you can see that there are a lot of CTS breast cancer publications, which is the green circles and bar at the top. There's also some help text when you hover over the question mark, which directs you to click View Summaries for more detail. So on this page, you can view summaries for each individual paper, and this is organized by topic. So for each publication, we can see the title, we can see the authors. There's also a brief summary of the study findings, and also a link out to PubMed to view the publication. There's also the option to view this by year instead of topic, if you wanted to, for example, view just the most recent publications. And then if we can click next again. Finally, I just wanted to show you what the back end of this visualization looks like. It's a really simple Excel sheet with those fields of interest, the year of publication, the topic, the title, etc. And what I want to leave you with is the idea that if your study has data that can be formatted like this Excel sheet, you can make visualizations like this and you can make them public. And if you're interested in doing so, we're here as a resource to help get you started and to share what we've learned. Thank you.
Thank you, Jim and Emma, for that presentation and that demo. I would now like to introduce Robert McInnes from the Cancer Council Victoria in Australia, who will also be presenting on a newly approved consortium project, the Population Attributable Fraction Project.
Thank you very much. Thank you for giving me the opportunity to present this new project. So one of the key strategies we can take for reducing the cancer burden is to target key preventable risk factors. So the population attributable fraction is a measure that can be used to rank and quantify these key preventable risk factors. Specifically, if the proportion of the cancers can be prevented if the exposure to the risk factors were removed or reduced. So there's been a few studies that have published these population attributable fractions, and they've either used published exposure associations or they estimated associations from cohort data. But using the published associations, however, have their limitations. So they include, assuming the independence between each of the exposures. The subgroup data is hardly, rarely available. It's not fully adjusted, and there's no interaction information, and they don't account for competing risks such as mortality, and then also they also have different lengths of follow-up. Then other publications have used, estimated the association from cohort data, but they also have their limitations. They include exposure prevalence from the cohort. It's not representative of the general population. And also, if they use the external representation of prevalence data, it's often not contemporary. And then while it's important to estimate the past burden, we feel that it's also very important to estimate the future burden of this cancer risk, particularly for policy making. And then they also don't account for the competing risks, and subgroup data is rarely available. And then also I also have to mention another limitation I haven't listed here is that most studies haven't addressed is not correcting for measurement error of the exposure. Can we have the next slide, please? So here I'm going to propose to undertake a population attributable fraction project within the Cohort Consortium. So that's led by Stephanie at Harvard University, Merritt and Claire at the University of New South Wales in Australia, and myself. Merritt, Claire, and I currently lead an Australian population attributable fraction project, which includes 7 studies within Australia. We have developed various methods to overcome many of the limitations that I described in the previous slide. And we produced several papers in the last few years on several outcomes, including colorectal, lung, endometrial, breast, pancreatic and kidney cancer. So drawing on our experience with these population attributable fractions analyses, we would like to expand on this area of research using a much larger resource within the Cohort Consortium, the Diet Cancer Pooling Project. Using this resource will really benefit, from using large international data to really test, for the strength of the associations and analyses. It will give us stronger evaluation of less common cancers. We'll be able to look at risk factor and interactions and subgroup analyses with much more power. And then we also pair this information with the country-specific exposure prevalence data. So our overarching goal is to estimate the population attributable fractions for cancer incidence and mortality, in particular, really the future burden of the cancer risk. Next slide, please. So the cancer outcomes we're interested are the ones that are listed here in the first column here, all the main common cancers, but there's also several rare cancers in here. The exposure of interest includes smoking, alcohol, body composition, physical activity, dietary variables, and women's reproductive factors. And then using such a large pooled cohort, we'll be able to have substantial power to do these subgroup analyses, such as by sex, age group, country, family history, educations, and so forth. And also we'll be able to perform formal tests for differences between these subgroups. Advantage of using the DCPP is that most of the data needed is already being or currently being collected or harmonized. And our plans for funding include the applying for the WCRF, which is actually due today, focusing on the cancers in bold there in this first column, because these are the ones that are the most up-to-date information in DCPP. We'll plan to apply for other funding in the future for the other cancers listed. So as mentioned, this project's being approved by the steering committee, and an abstract is in the meeting website, and I think details are going to be further circulated in the next newsletter. So please contact us if you're interested in participating in the project. Okay, thank you.
Thank you, Robert, for your presentation. I would now like to introduce Mahdi Sheikh from the International Agency for Research on Cancer, IARC, who will present on the Opioid Cohort Consortium to investigate the effects of regular opioid use on cancer development.
Thank you very much, Camille. And hello, everyone. Thanks for the opportunity to present our planned consortium. Next slide, please. So this consortium is initiated to address a growing global health concern that is unfortunately affecting many countries, and that is the overuse of opioids in the world. The short-term health consequences are well known. Preventive policies have been taken by many countries, but unfortunately the long-term health consequences are still unknown. So here I'm showing the scientific evidence on the association between opioids and cancer risks. As you know, opioids are categorized into two big categories of opiates, which are the natural opioids like opium and heroin, and also pharmaceutical opioids. These two categories have some shared contents and also some shared pathways. There has been a lot of evidence on the association between using opium and has been lots of evidence on the association between using opium, and also associations have been shown with at least 9 types of cancers in human studies and also experimental studies. So these evidence were evaluated recently by the IARC Monographs program, which resulted in the classification of opium consumption as group 1 carcinogen to humans. On the other hand, when we see pharmaceutical opioids, there is paucity of evidence there, and most evidence in human studies arise from registry data linkage, which have consistently shown associations with several types of cancers like lung, urogenital, and liver cancers. And there have been many experimental studies showing the association of opioid use with some of the opioids with genotoxicity, and also tumor promotion effects like immunosuppression and neovascularization, and also studies have shown the presence of opioid receptors on some cancer tissues and their involvement in tumor initiation and progression. As you see, there are many limitations in the current evidence, specifically from human studies, as confounding effects cannot be ruled out, and also there has been no large-scale cohorts seeking this association, most probably because opioid use data have not been gathered in most cohorts, and cohorts with opioid use data have very limited numbers to provide the power to assess these risks. So comprehensive consortium-based approach is obviously needed. Next slide, please. So this is an overview of the Opioid Cohort Consortium, Opico, as said earlier by Dr. Ginsberg. This consortium was approved for grant support by the International 100K Plus Cohort Consortium. The overarching aim of this consortium is to build a strong international resource for multidisciplinary scientific studies on the use of opioids and their long-term health consequences. The main exposure is the use of any types of opioids that has been gathered through questionnaires on natural opioids, or on the medication use data upon enrollment, or through linkage to some national records, like national prescription records. And the main outcome that we are interested in is cancer analysis and also mortality analysis, all types of cancers, and also digestive, respiratory, urinary tract, and brain cancers that we found to be associated with natural opioids in our previous analyses. So the aim and approach, we aim to organize data on opioid use from large-scale prospective studies. As you saw, opioid use data have been gathered through different ways. So to compile new data on opioid use in large cohort studies through linkage to national registries when applicable, and also to assess the types and trends of, and distribution of opioid use across diverse population, and to determine the association of opioid use with cancer incidence and mortality. But also many of the involved cohorts have got like some genetic data. They have got like some biospecimens, so it will, harmonizing opioid use data will allow to be used for these types of analysis, biomarker and genetic analysis. So will be a very rich consortium. So next, please. So we're inviting all cohorts who are eligible to participate in this study. Up to now, more than 8 cohorts have initially agreed to join the Opico, with more than 1,700,000 participants, with 80,000 opioid users. So cohorts to be eligible, they need to have opioid use data through questionnaires or through data linkage. So any cohort that has gathered medication use on enrollments would be eligible. So the outcomes, death and cancer, demographics, if they have data on demographics, smoking, alcohol intake, and chronic health conditions. This consortium is currently led by IARC Genetic Epidemiology Group. Dr. Paul Brennan, Dr. Hilary Robbins, and myself are coordinating this consortium, and here is the contact information. So please join us. Thank you very much.
Thank you, Madi, for your presentation. I would now like to introduce our final speaker for this session, Deirdre Tobias from the Brigham and Women's Hospital and Harvard Medical School, who will be presenting on metabolomics of obesity and obesity-related cancers.
Thank you, Camille. I'm really excited to present this potential collaborative opportunity to you all today. I'm an obesity and nutritional epidemiologist at the Brigham, and I'm increasingly interested in the role that obesity plays in developing cancers downstream, but more proximal to the development of obesity and weight gain, initiating the cancer itself. I see metabolomics as one potential tool to elucidate this. As you are all probably very much aware, excess body weight, including obesity, is a risk factor for at least 13 cancer sites and is estimated to contribute to approximately 9% of the cancer burden in developed countries. And in the US, overweight and obesity-related cancers comprise over 50% of cancers in women and a quarter of those in men. The mechanisms linking obesity with cancer generally have mostly been investigated in a site-specific manner. For example, sex hormones playing a role in reproductive site cancers, inflammation, adipokines, lipids, oxidative stress, all of these mechanisms that get disturbed with weight gain and obesity play some role or another in a variety of cancer sites. The human metabolome, on the other hand, I think is still very much unexplored with its impact or its contribution to obesity-related cancer risk. And I see this as potentially a marker for site-specific cancers, but also for looking at obesity-related cancers overall as a composite endpoint by being able to investigate more proximal to the weight gain upstream of cancer development, what might be related and therefore prevented for cancer risk. So could you please go to the next slide? Thank you. So one set of aims that could leverage this, this hypothesis to look a little bit further at the role of metabolomics is to first prospectively evaluate among cohorts that have, for example, baseline blood samples or perhaps already metabolomics assayed for other projects, look at previously identified obesity-related metabolites. So metabolomics is widely done in cardiometabolic, diabetes, and obesity-related chronic diseases. And to that end, there have been a number that have already been implicated with obesity and insulin resistance and cardiometabolic disease. So looking at these candidate metabolites in relation now to obesity-related cancers is one approach. And then the second might be to identify novel obesity-related metabolites, just looking, for example, cross-sectionally with anthropometric measures of body mass index, which is a marker of total body fat or waist circumference, which better captures central adiposity, which is often more implicated in cardiometabolic disease and therefore might be relevant to some obesity-related cancers in particular. And then look at those metabolites that are identified de novo for their long-term association with incident obesity-related cancer risks. And I think this catch-all term of obesity-related cancers is quite broad and can include additional sites as more associations are established. And ultimately, this might help inform a precision prevention goal. If metabolites underlying these relationships can be identified, then that might help inform these blue bars here, which are ways in which we might be able to intervene to regulate the metabolite levels. So these are circulating biomarkers that are the result of some process or another, whether it's lifestyle or diet or medication or metabolic state. And if we can leverage knowing someone's metabolomic status, if we know how to change that and then subsequently reduce downstream cancer, that I think would be part of the ultimate goal from this. And then the final slide, please. So one example is an analysis that we conducted just in the women's health study, but I think is an example of something that can be analyzed across cohorts to improve statistical power. That would be the ultimate goal, I think, and also generalizability across platforms and population characteristics. But for this analysis, we looked at, so these yellow bars here in this table are just an example of a variety of metabolites and their pathways that have been implicated in obesity. So these then could be looked for cancer associations. These are 26,000 women in the women's health study, which is a US cohort. The mean age was about 55 at the baseline blood draw. So these are NMR spectroscopy quantified branched chain amino acids. And branched chain amino acids include isoleucine, leucine, and valine. They're essential amino acids, meaning they're derived solely from diet. However, diet does not strongly correlate in indicating their metabolism, and downstream breakdown is probably more responsible for the levels that we see in circulation. Because of their strong relationship with obesity and type 2 diabetes in particular, they were a candidate that we wanted to look at for this composite endpoint of obesity-related cancers. So after 24 years of follow-up, there were 2,700 of these cancers, which were about half of the total cancers in this study population, and we adjusted for a number of potential risk factors at baseline. as you can see, contrary to the hypothesis that I came in very excited with, there was no association between plasma branched chain amino acids across quintiles here with risk of total obesity-related cancers. Maybe some suggestion of association for leucine when we evaluated it continuously per standard deviation, and the p-trend, is borderline there. Excluding non-fasting samples, as I mentioned, branched chain amino acids have somewhat of a metabolic correlation. Actually, some did strengthen these association or the association for leucine, but still not as strong as the association we see for something like type 2 diabetes. So in the same cohort, looking at baseline branched chain amino acids with incident type 2 diabetes over 25 years, these hazard ratios out in Q5 are 2 or 3. So they're much larger and indicative of something going on there. I don't think that's the case, particularly for branched chain amino acids globally in this composite endpoint. So looking more site specific could be the next step, which I think some colleagues are looking at. So this is just, again, a topic that I think consortium can contribute to, because metabolomics are expensive and often done in a case controlled type manner, and pooling across sites I think could also be telling, at least for obesity, as an upstream exposure for a number of cancer sites. So thank you.
Thank you, Deirdre, and to all of the speakers. I will now pass things over to Heather, who will help to lead the discussion for the session.
Great, thanks so much, Camille, and thank you to all of the lightning talk speakers. Really appreciate the insight and inputs and future projects that are very exciting. For the rest of you, please post any questions that you have into the chat, and we will try to follow along with that. But I will get started with a few questions to start with. So, I think this is it's a great collection of potential projects. We've got 2 newly approved Cohort Consortium projects with Andriana's and Rob's projects. And then I think with Maddie and Dee Dee's projects, those are examples or potential examples of cross-consortium projects that sort of intersect a couple of possible consortia, which are also interesting possibilities. And then Jim and Emma, it was great to see the tools that you've implemented in CTS that may be applicable to other cohorts. So I wanted to start with Andriana and ask a question about your a proposal to match not only non-cancer controls to the appendiceal cancer cases, but also to match some colorectal cancer cases as well. And I was wondering, in the work that your group has done previously, have you been able to observe similarities or differences between appendiceal cancer risk factors and CRC risk factors?
Yeah, sure. So great question. Thank you. On the risk factor front, no risk factors for appendiceal cancer are currently known, with the exception of a study using National Health surgical quality data that looked at risk factors for patients diagnosed with appendiceal cancer among those undergoing appendectomy. So that's one of the real novelties of this project, is to really make significant advances in understanding risk factors of appendiceal malignancies. In terms of molecular features, emerging evidence is suggesting that there are distinct molecular features from targeted sequencing data between colon cancers, particularly right-sided colon and appendiceal malignancies, to suggest potential distinct biomarkers and etiologies. Of course, given the lack of robust data for appendix cancer at present, treatment regimens that have been recommended are extrapolated from studies from colon cancer. So this also ranks as an important area for further investigation in terms of appendiceal cancer-specific biomarkers, given what's emerging as distinct molecular features of disease.
Great, thanks so much. Jim and Emma, a question from Garnet. And Jim answered the question about what technology platform was used for the visualization and about compatibility. So Jim has posted that into the chat. And I had a follow-up question. For many of us, seeing these very slick visualizations is so attractive to being able to visualize the data that we've collected. And I know for many cohorts that have collected data over time, legacy data is such a big hurdle. And so I wondered, as you got to this point, how much for CTS was the effort going into cleaning and preparation of the data versus work developing this particular data visualization platform?
It's a good question, and let's see how to tackle it. For the CTS, we really started with using Tableau internally. There's a licensed version that we use for a lot of our internal analyses, and then we discovered the Tableau public version, which is free and presents summary data. And what was particularly appealing about sort of a Cohort Consortium perspective is for all of these projects, whenever we would get to the point of answering one of you, our colleagues' questions about how many cancers we have, how many biospecimens, whenever we answered that question, we said, all right, that's a trusted source of data. And then we started to think, can we put that level of sort of cleaned, presentation-worthy data into the Tableau public environment? I put some links in the chat there. Emma, anything you want to add on that?
No, I think that pretty much covers it. I would say in our case, the majority of the time was spent making sure that there was no data that shouldn't be exposed, since this was kind of a new realm for us, making sure that everything was aggregated and summarized to a level that it was not identifiable was probably the most time-consuming manipulation of the data.
Great, thank you both, and thanks for putting the links in the chat. So a continued question on the intersection between consortia. So Didi, Mark Gunter has asked if your project is linked to the COMETS Consortium, and I was also going to ask the same question. I know there's ongoing work there to identify obesity-related metabolites. I think many of us are cross-linked between these two consortia as well. So that might be a good place to pursue.
That sounds like a great opportunity. So I'd love to get further engaged with the COMETS Consortium. I know it does obesity, diabetes, and a number of cardiometabolic diseases, right? So, but I, to date, have not been engaged. So that would be, it would be great to include it and begin that collaboration.
Excellent. That sounds good. And Maddie, I had a question for you on the Opioid Consortium. And again, this is an intersection between IHCC that we heard about from Jeff Ginsburg and the potential of leveraging additional cohorts through the NCI Cohort Consortium here. And I was wondering about data collection on opioids. You talked about using registries and reported medication use, and I'm wondering about illicit or recreational use of opioids and how or what you can do about capturing that as you're looking at these relationships.
Yeah, thank you very much. So there are some cohorts because we have some cohorts that have gathered opium use data, specifically Asian cohorts. So they are already involved here. And the main concern that we really didn't have any cohort analysis was the opioid prescribed opioid use data. So we are using two things. First, some cohorts did not have this data, but now we have very good national records for merging this data. We are trying this approach now with some cohorts in Australia and also in Scotland. So trying to harmonize the data like the self-report medication use data. And also we're inviting, we have now with some recreational cohorts with the recreational use data 30,000 people, and also with opioid use data 40 to 50,000 people. So we have both categories and we're trying to look at both categories, but we are inviting cohorts if they have this data to join us.
Wonderful, thanks Maddie. And I had one last question for Rob concerning the PAF analyses and using the Diet and Cancer Pooling Project. And I was wondering, you mentioned that many of the variables of interest are already in those collected consortium data. And I was wondering, how much can you get started on using the existing data and what additional types of variables would you like to have added to the DCPP to enable the PAF project?
Yeah, so I think really, we have the data pretty much harmonized, and I think we initially chose those variables and the cancer outcomes because they're kind of the most up to date. But I think the most pertinent thing is to try and get most up to date information. I suppose, for the cancer outcomes, that's kind of that will be kind of like the key going forward, especially for some of the other, less common cancers, to make sure that, the follow-up information is completely up to date. I think that's pretty much our focus.
Sounds great. All right, well, thank you all very much for giving us your lightning talks. It's been a great, quick ride through some of these projects that are getting off the ground, and thank you very much for sharing them with us. So that will conclude our session 2.